Welcome to Học Tiếng Anh với Joseph, where we build vocabulary and practice pronunciation through reading. Continuing with The Cat in the Box, A History of Science in 100 Experiments, today we read story number 80. Hôm nay chúng ta sẽ đọc câu chuyện số 80, Jumping Genes. Until the late 1940s, everybody knew that genes were stable entities strung out along a chromosome like beads on a wire. That image was challenged by Barbara McClintock, working at the Cold Spring Harbor Laboratory in New York. But it took a long time for the significance of her experiments to be widely recognized. McClintock started her research at Cornell University, New York in the late 1920s, using studies of maize plants, sweet corn, to build upon the pioneering work of Thomas Hunt Morgan with fruit flies, in an echo of the way in which Gregor Mendel had studied heredity using pea plants. She developed techniques to stain the cells of maize plants so that the 10 chromosomes they contained could be clearly distinguished using a microscope. And with the assistance of Harriet Creighton, she was able to show that specific changes in the chromosomes were associated with specific changes in the plant itself, in the phenotype. It was possible to identify individual genes within the chromosomes and to compare the chromosomes of one plant or one cell with those of another. In one example, a maize strain being studied carried chromosomes that were associated with either light or dark kernels in the corn cobs. When the cells were stained and studied under the microscope, the researchers noticed that the difference was associated with a difference in chromosome number 9, with a chromosome in the dark variety having a visible knob that was not present in the equivalent chromosome of the other variety. Those results were published in 1931, but the experiments took a long time to develop further. For the same reason that Mendel's experiments took a long time, the slow rate at which plants reproduce, compared with things like Drosophila fruit fly. The advantage is that instead of having to catch flies an eighth of an inch long and look at the color of their eyes, you can simply peel back the leaves of the corn cobs and see the pattern of colored seeds lined up for inspection. Wild maize, unlike the kind sold in supermarkets, has multicolored cobs. McClintock patiently continued her studies, moving to Cold Spring Harbor in 1941, and made her most important discoveries there in the years following 1944. In order to study the genetics of maize, McClintock did not need to know whether it was protein or DNA that carried the genetic information. All she had to know was that the information was carried on the chromosomes and that each chromosome is made up of separate genes that carry specific instructions for the life processes of the plant. McClintock was studying how mutations, changes in the genes, affect the pattern of pigmentation in the corn, how the genotype affects the phenotype. In some plants, she saw splashes of the wrong color on some of the leaves, such as a streak of dark green on a light green leaf, because the leaf grows by the repeated division of cells. she could trace this mutation back to the very cell at the start of the streak of color in which the mutation had occurred. In some cases, these multicolored patches developed at a different rate from the rest of the plant, either growing more quickly or growing more slowly than their neighbors. After years of painstaking research involving generations of controlled crossbreeding of her plants, McClintock was convinced that what she was seeing was evidence that the behavior of some genes is controlled by other genes. In this specific example, one of the control genes sat next to the genes responsible for the coloring of the leaf on the same chromosome and turned it on or off. But a second control gene affected the speed with which this process occurred, and that gene did not have to be on the same chromosome. Finally, having identified these genes, she found that the second gene does not even have to stay in the same place. It could get moved around during the process of self-division, so that by comparing different cells, it looked as if it had moved from one place to another within a chromosome, or had even jumped from one chromosome to another, the process she called transposition. This flew in the face of the established idea of the genome as a fixed set of instructions passed unchanged from one generation to the next. McClintock summarized her work in a scientific paper titled "The Origin and Behavior of Mutable Loci in Maize," published in 1950. It eventually changed the way scientists think about genetics and inheritance, and it opened the way to an understanding of how genes regulate the production of proteins in the body and to developments in genetic engineering. In 1983, at the age of 81, McClintock belatedly received the Nobel Prize in Physiology or Medicine for her discovery of mobile genetic elements. To the vocabulary section. Chromosome, beats on a wire, beats on a wire. Challenge, distinguish, phenotype and genotype. Kernels in the corn cobs, mutation, pigmentation, crossbreeding, transposition, flew in the face, fly in the face, flew in the face. Thank you so much for your time, and I will see you in the next story. Bye for now.